Hello, everyone, and welcome back after the short break. We're moving on to our next session now, which is the session focusing on our BRC novel therapies theme. And I'm delighted to welcome Dr. Karin Straathof, who's one of the deputy theme leads for this theme, to chair this session. Thank you, Karin.
So thank you for the kind introduction, Laura. It's a real pleasure to chair this session, showcasing the work of the Advancing Novel Therapies theme. In this team, we build our capacity and accelerate the development of clinical trial options for children who are affected by rare and complex conditions who have currently limited treatment options. Like the previous sessions, we will have 3 speakers, and together they will give you an overview of the many novel treatments that have been developed over the past years, and they will show you how they have improved the lives of children with rare diseases. So the first speaker in this session is Professor Francesco Mattoni, lead of the Advancing Novel Therapies team. Professor Mattoni is a pediatric neurologist and an international expert in the pathological and molecular aspects of neuromuscular disorders. And he leads a clinical research program for children affected by neuromuscular disorders. Professor Mattoni will give us an overview of this BRC team entitled Learning from Rare Childhood Disorders. Thank you, Professor Mattoni.
Thank you very much, Karin. And let's see if this works. This work for everybody so far? And it should work for me. Excellent. Well, thank you. I'm going to take you through what our theme does. I also introduce you to Manju and Darren, who will be the team leads for the next round of the BRC. I will talk is a high-level talk on what is the Advanced Novel Therapy theme, how we spend the money in our theme. I will make a specific example of benefit for patients that we've been able to achieve in the last few years. I will also stress how funding helps keep people in training, and also I will talk about how our experience in the last few years is taking the foundation of the next application for the BRC. The main areas where we fund activities are those of the complex clinical trials in childhood rare disorders, in developing novel therapies. We have a biomarker discovery, a number of projects there. We have a yearly research funding calls that has been quite effective. And we also have been involved in training the next generation of translational clinical scientists. And the target disease areas are inborn errors of metabolism, inflammatory diseases, multi-drug resistant bacteria, neuromuscular disorder, neurological disorder, and respiratory condition. And I will start from probably one of the areas where there has been more advance in the novel therapy regarding at least the RNA therapies, but also the AAV gene therapy has been on spinal muscular atrophy. This is still a inverted comma rare disorder, although 80 children are born in the UK every year. And this is the most common lethal genetic disorder in infancy. The most common form, type 1 SMA, you can see an example on the right-hand side of your screen. These children will not survive past the age of 9 months. And there have been 3 drugs, all studied at the Thornton Street Hospital, an antisense oligonucleotide, an AAV gene therapy, and recently a small molecule that have been studied and approved. And I think just to give you a visual example of what these drugs can achieve, on the left-hand side, you see a paralyzed little infant. And this is the same child after 2.5 years of treatment with this drug, Spinraza. And clearly, this is spectacular because by that time, this child should not be alive, should not be breathing, and certainly would not be speaking and standing. I think, however, more recently, we really realized that the pre-symptomatic treatment will make an even bigger difference. And this is the example of a child that has been treated pre-symptomatically this is the little girl, the blonde girl, and the other girl in the wheelchair is the child who is the older sister, who unfortunately at the time could not access any treatment. So this is where we are aiming to. And I think one fair question is, what is the added value of the BRC investment for studies like this? I have stressed these are high intensity studies, but what else does the BRC do in addition to provide a safe place to do this clinical trial? I have used again a few examples. I think that we have a BRC supported national registry where we collect information of treated patients nationally, and this is being used for NICE and NHS England to manage access of these drugs. We also, in collaboration with national and international cohort of patients, a cohort of patients and clinical investigators, have identified areas where the evidence for broad use of this drug were lacking, and we have generated this evidence that led to an increase of the inclusion criteria in the NICE program. As I indicated in the previous slide, newborn screening and early intervention is necessary to get the best out of this drug, and the BRC has funded a pilot study to assess the feasibility of this in a diagnostic setting. We are studying the feasibility to recognize better and worse responder using a number of biomarkers. The work that we are doing has allowed us to leverage external funding from the European Commission, and we have done biodistribution studies that are really the only example of in vivo biodistribution study in people receiving this AAV. Another striking example is the one from the Batten disease. This is also a very severe condition where, in addition to the motor decline that this patient had, and within a few years from onset, this is at the age of 3 years, this is the same child at the age of 5 years, so a normal child will lose the ability to walk, but not only that, will develop epilepsy and also progressive intellectual disability. A drug was trial at Greater Ormond Street Hospital, and actually, this is a drug that is delivered as an intracerebroventricular administration, and it was able to stabilize the condition, and this became available in the country. But there is a problem that once you give this drug intracerebroventricularly, it does not reach the retina, and visual loss is a feature of the condition. And that again led my colleagues for Thyssen, together with the ophthalmology team, to initiate, with support from the BRC, a compassionate use of the intraocular administration of Brineura. And this is a study that is going on in collaboration with the advocacy group. An example of drug discovery comes from the group that has been addressing the issue of antibiotic resistance, and in the work of Professor Callaghan, together with other colleagues at UCL, was able to identify a drug that has actually been repurposed from a different indication, and this drug properties appear to indicate that a new class of antibiotics may have been identified. I think the work that the colleagues have done was that of improve the characteristic of the original compound, and if you like, a strong activity against the Mycobacterium abscessus was achieved with a good resistant profile and IP protection. And also very interesting example of the biomarker discovery group has been from the group of the Translational Mass Spectrometry Research Group, and an example of a discovery using mass spec for a relatively rare group of conditions is on the top, where new biomarkers for cardiac disease progression were identified, patented, and now used by industry. But I think it's, during COVID, some of this expertise was also used to identify methods to detect antibody response and immunological response, for example, to the different variant of COVID-19. Not only that, the same group of colleagues, but this time in collaboration with a number of other UK healthcare providers, were involved in the operation Moonshot, where the mass spec was used, this time not to look at the immune response against the COVID, but actually to identify the COVID antigen presence. This was experimented and very rapidly led to a kit that is now available and is distributed. And I think the timeline in which this has been developed is astounding. Very briefly to say, Amy spoke before, and I think that my point here is on how the investment of the GOSH that is indicated every time you see a red square means that there has been some BRC involved funding, either via catalyst funding and or also with additional research funding, and how this has helped in to get into the MRC Clinician Scientist Fellowship. And very similar, Julian, who also was another BRC Catalyst Fellow, moved from, if you like, really a postdoc in an early translational path, but to be able to have his own research group. These are my last slide, and is to say that the antisense oligonucleotide have really been heavily used in the BRC in the last few years with spinal muscular atrophy and delivery intracellular ventricularly, and the Duchenne muscular dystrophy with intramuscular delivery. A number of drugs have been approved, but I think the expertise has led now to fund a group of, to identify a group that will take forward each of these different indications that I will not necessarily mention one by one as areas of development. Patent have been filed in this area, and it is very likely there will be a large number of new academic-led clinical trials in this area. This is my last slide, and is really to show that there have been activity regarding approval of other drugs for rare disorders. I indicated that the GOSH BRC is involved in the next generation therapies, and also we train colleagues, not only from the medical background, for being able to develop and take forward translational research. And I thank you for your attention, and I'll close my screen.
Thank you very much, Professor Montoni, for a beautiful overview. Very much enjoyed that. We will now move to the second speaker of this session, and I have the pleasure to introduce to you Professor Helen Cross. Professor Helen Cross is the deputy lead of the Novel Therapies team. She is a consultant neurologist and head of the Developmental Neurosciences program at the Institute of Child Health, and she's also the current president of the International League Against Epilepsy. Helen Cross will share with us her work on novel therapies for childhood epilepsy. Thank you very much.
Thank you very much, Karin, and I hope everyone can now see my slides. So, yes, I'm a professor of childhood epilepsy, consultant paediatric neurologist at Great Ormond Street Hospital, and also have been the deputy theme lead, particularly with the CRF high intensity, the inpatient trials for the BRC. But what I'm going to focus on is particularly where we've moved forward with regard to novel therapies for the complex childhood epilepsies, and particularly focus on one area where there's been a Pan-London and industry collaboration resulting in a treatment to provide an easier form of treatment from a traditional one, which I'll explain. So epilepsy, a disorder of the brain characterized by an enduring predisposition to generate epileptic seizures. Traditionally, we utilize anti-epileptic medication, but 30% of the individuals that we treat, particularly the children, remain resistant to this medication, and this proportion has not changed in 30 years, as illustrated by the study on the right. Many, as Amy pointed out earlier, in this group have early onset seizures and have an eye association with poor developmental and behavioural outcome. And increasingly, as we get more in the way of diagnosing the cause of the epilepsy being the symptom, we recognise this as a group of rare diseases, the epilepsies, for which we should be targeting our treatments more effectively. First, I'd just like to focus on two novel therapies that we were in collaboration with the industry pharma companies that were developing these compounds, were able to collaborate in developing the clinical trials and providing the data to bring them to the regulatory space and make them available for our children with particularly rare and complex epilepsies. Firstly, cannabidiol. Many of you will have heard in the press about cannabinoid compounds being a possible treatment for complex epilepsy. GW Pharma have been interested in the utilization of pure cannabidiol since 2007 in the treatment of epilepsy and produced a pharmaceutical preparation, Epidiolex. I was involved in the initial treatment of a sentinel case in 2014, which showed a dramatic reduction in seizures, and this allowed an IND in the USA, which allowed an open label study across 6 centres to demonstrate some benefit, and was also involved in the development of the randomised controlled trials in Dravet and Lennox-Gastaut syndromes, particularly difficult rare syndromes of epilepsy, and also Finborough-Callahan, my colleague, in the trials with regard to epilepsy associated with tuberous sclerosis. These demonstrated benefit, bringing data available for regulatory approval both by the FDA and the EMA, and cannabidiol is now approved through NICE HTA for utilisation in Dravet and Lennox-Gastaut syndromes in association with clobazam. Fenfluramine is a slightly different story. This was originally a medication used for weight loss in the 1960s. It went out of favour because of cardiac problems, but was also demonstrated to be effective in reflex epilepsies. And ultimately, through evaluation of certain patients that were utilising this medication in Belgium, found specifically to be effective in Dravet syndrome. We were involved in the development of the randomised controlled trials in Dravet syndrome, and also thereafter having recruited and published the data required for the regulatory approval in the FDA and the EMA, and it's currently under evaluation from NICE with regard to an HTA. But what I want to concentrate on this talk is really moving forward an old treatment. The ketogenic diet is a high-fat diet that was designed to mimic the metabolic effects of the body on starvation. Through the breakdown of our own body fat, we produce ketones, and if we take fat as our main energy source, we also generate ketones. This was discovered in excess of 100 years ago to be effective in epilepsy at a time when we did not have anti-epileptic drugs. There are different ways we can administer this diet, either using long-chain fat and low carbohydrate, low protein, the classical way, utilising medium-chain triglyceride, this being higher in generation of ketones per calorie than long-chain fat, the MCT diet, or a modified ketogenic diet many of you will know as the modified Atkins diet. And although traditionally it was felt to be unpalatable, there are many ways we can now give this and make it more palatable, even in a tube format. We demonstrated in 2008 with the publication of a randomised controlled trial that the ketogenic diet was effective over and above no change in treatment in children with complex drug-resistant epilepsy, and there have been further randomised controlled trials demonstrating this efficacy. However, the million-dollar question has always been, how does this ketogenic diet work? The presumption has been ketones, but it's not ketones necessarily that are produced being an anti-epileptic drug, and there are many other pathways on which the ketogenic diet may have an effect. And maybe there will be different mechanisms from different diseases, which makes it more complex when trying to divide whether there is a simpler way to giving this treatment other than completely changing the diet and restricting what a child or an adult eats. One particular thought with regard to this was developed in parallel across London. Robin Williams and his team, in a collaboration with Matthew Walker at the Institute of Neurology, Robin Williams at the Royal Holloway, were looking at different compounds with regard to reducing epileptogenicity in rat brain slices, and determined that decanoic acid, C10 fatty acid, was particularly effective and even more effective than perhaps some of the standard compounds such as sodium valproate. In parallel, Simon Heels and his team at the Institute of Child Health have been evaluating the possible effect of C10 on mitochondrial activity as a possible mechanism of action of the ketogenic diet. And in neuronal cells, demonstrated that by treating with C10, there was an increase in citrate synthase activity, a marker of mitochondrial function. And indeed, looking at numbers of mitochondria per cell, also an increase seen, as seen on the right here in an electron microscopy of a cell. Some of our clinical work also suggested that mitochondrial function may play a part in that carnitine levels at baseline prior to starting a ketogenic diet was significantly higher in responders than non-responders. And carnitine required for uptake of long-chain fat, particularly for utilization in the mitochondria. Further work has then confirmed the possible anti-epileptic effects of C10 decanoic acid, demonstrating a similar mechanism to one of our standard anti-epileptic drugs, perampanel, as a non-competitive AMPA antagonist. So having this evidence base that C10 fatty acids may be effective or may be a very effective component of the ketogenic diet, bearing in mind that actually our traditional MCT that we were utilizing was predominantly C8, in collaboration with Vitaflow International, a nutritional company, a predominantly C10 compound that was palatable was produced, and this was 80% C10, 20% C8 for stabilization. And we undertook a tolerability study in both children and adults across UCL. And here we were aiming for recruiting 40 to each. The compound would be taken 3 to 4 times daily with food, a normal diet maintained. And in this study of individuals with drug-resistant epilepsy, we were able to start 61 on the compound, and at the end of a 3-month period, 41 remained on the compound. In effect, it was very well tolerated, although there were some gastrointestinal effects that could be adjusted by adjusting the rate of introduction and indeed the proportion. Of course, although we were looking at tolerability, we were also looking at seizure response. And even though we didn't have a baseline seizure frequency, by measuring the seizure frequency over the 3-month period, despite the fact we only counted seizures from the start of the treatment, there was an overall reduction over that 3-month period in both adults and children with regard to seizures. And measuring decanoic acid levels in the blood, there was a correlation with the amount of C10 with the amount of seizure reduction. So moving forward, this compound then went forward for approval as a nutritional compound from the Advisory Committee on Borderline Substances, a committee that assesses nutritional and dermatological substances that don't fulfill complete criteria for medicinal compounds. And having got that approval, this can now be used more widely in individuals with epilepsy. We're also in development of a protocol for evaluation in a particular metabolic disorder that responds to the ketogenic diet, GLUT1 deficiency, and we did have a particular effectiveness in individuals in our study who actually have this disorder. We've been evaluating with patients and the public with regard to what will be required for such a study, and not looking at what would be meaningful outcomes and indeed the maximal duration of such a study. So that just summarises where we've come. It was a first-in-man treatment. We move forward to try and make an easier way to administer a very old treatment and make it much more widely available to our patients with complex epilepsy. As we move forward, we're going to look further at the cannabinoids, not least more broadly than cannabidiol alone, looking at the role of THC and is it safe. I'm doing this in collaboration with co-chief investigator Finbar O'Callaghan at the institution and in discussion with the NIHR and NHS England. And as Amy illustrated earlier, we're characterising the rarer epilepsies, looking at etiological targets, moving towards precision medicine and even genetic therapies that are currently being evaluated. And overall, therefore, we're able to improve not only the seizures, but all the other comorbidities that we see in our children and improve outcomes overall. So with that, I just want to acknowledge everybody who's involved in the K-Vita study and the development of C10 in its journey. And thank you for your attention.
Thank you very much, Professor Cross, for a fascinating talk. I very much enjoyed that. We'll now move to the third speaker and I'll have some questions at the end of the session. Our third speaker is Dr. Virginie Mario. She is a research associate in the Developmental Neurosciences Department at the Institute of Child Health, and she will give us a lightning talk to share her work on AAV-based therapy for cardiomyopathies. Thank you, Virginie.
Good afternoon. Thank you, Karin, for this introduction. So hypertrophic cardiomyopathy means a thickening of the heart muscles and is usually caused by a mutation. It is the most common inherited cardiovascular disease with a prevalence of 1 in 500 of the population. It has been estimated that children account for about 10% of the cases. Hypertrophic cardiomyopathy is usually passed down through families, hence the origin of this word inherited. If you have a parent with hypertrophic cardiomyopathy, you have a 50% chance of having the genetic mutation for the disease. To date, the only curative treatment is heart transplant. Our project was to establish the proof of principle that an AAV-based therapy can be used for cardiomyopathies. In collaboration with Perry Elliott and Paolo De Copi at Gauche ICH, from patients with inherited cardiomyopathies, we obtained skin biopsies from which dermal fibroblasts were expanded. Cells were reprogrammed into pluripotent stem cells and eventually differentiate into cardiomyocytes. At the lab, we expanded, generated, differentiated, and characterized these iPS cells. The successful reprogrammation was demonstrated by the expression of cardiac proteins by immunofluorescence staining, as shown here, such as encadherin, here in green, desmoplakin, here in red, or plakoglobin, here in green. And also, very important characteristic, by the capacity of the cells to be. Next, we designed AAV-based therapies for different cardiomyopathies. It has to be known that AAV vectors are non-pathogenic. They don't integrate into the host genome DNA, and they can be directly delivered by different injections to the heart. AAV vectors have been successfully used to treat cardiomyopathy in animals affected by several muscular diseases, including limb-girdle muscle dystrophy or Duchenne muscular dystrophy. In our first step of experiments, we transduced iPS-derived cardiomyocytes with AAV vectors coding the GFP protein. Different capsids were assessed, AAV1, 6, or 9, and we observed that AAV6 was the most efficient. This was the proof of principle demonstrating that AAV vector can be used in vitro on cardiomyocytes. However, one of the major bottlenecks of AAV vectors is their small packing capacity, which is limited to 5 to 4.7 kb. And for some of the genes we are studying, it is an issue because they are larger than 5 kb. So we design alternative strategy like the overlapping dual AAV strategy that is based on the co-transduction by complementary AAV vectors. The first vector carries a promoter and the 5' half of the genes, and the second vector includes the 3' half of the genes and the polyadenylation signal. The two vectors share a common sequence that is used for homologous recombination to reconstitute the full-length coding sequence. We have developed variations of this strategy, which, for example, include a spliced donor site in the first vector and a splice acceptor site on the second vector. leading to the concatenation of the two vectors at DNA level and the removal of the supplementary ITR during the transcription process, thanks to this splicing event. And it eventually allows the reconstitution of the full-length coding sequence. To conclude, this project is a nice illustration of a translational medicine where a loop from bench to bedside is possible. Indeed, from iPS cells derived into cardiomyocytes from patients, we aim to treat it with a dual AAV-based therapy that can directly benefit patients. This project was achievable thanks to funding by the BRC and the involvement of all these partners, whom I would like to thank. Thank you very much.
Thank you, Virginie, for a beautiful talk and explaining these novel treatments. And I very much enjoyed this excellent session, thanking all three of our speakers, really giving us a snapshot of the huge amount of activity in developing novel therapies and bringing these from the laboratory to our patients in the hospital. And with that, I hand back to Laura, who will take us to the next part of the program this afternoon.
Thank you very much. Virginie, if I could just ask you, there we go, to stop showing your slides. Thank you very much. Moving on, we're just going to finish off this session by showing you a brief video which was created by our research comms team led by Lauren Tadaldi, showing us really some of our patients just making some comments. And this video was made for Rare Disease Day last year to celebrate Rare Disease Day.
Hello, my name is Lucrezia. I'm 7 years old and I have a rare disease named peripheral neuropathy.
I'm a pediatric neurologist working on rare neuromuscular conditions at Great Ormond Street Hospital. The 100,000 Genome Project allows us to give to Lucrezia and her family a final diagnosis, ending a long diagnostic odyssey.
My work as a senior nurse has been looking at how juvenile dermatomyositis makes children and young people feel and how we can help support them better.
JDM left me too weak to walk or even feed myself, but thanks to research at GOSH, I am now in remission on medication.
Research helped me.